Lecture 10, Module 1, Inducing BAT Thermogenesis, presented by Sarisha, Stephen, Vivek, Kevin, and Allison. Learning objectives. Inducing UCP1 leads to increased metabolic rate. UCP1 deficient mice are cold sensitive. Activating BAT activity through cold exposure increases triglyceride clearance. B3AR agonists stimulate BAT. And capsinoids induce thermogenesis. Thermogenesis is the production of heat in an organism's body. There are two types of thermogenesis: shivering thermogenesis, which occurs in the muscle, and non-shivering thermogenesis, which occurs in brown adipose tissue, BAT. We are going to focus on non-shivering thermogenesis in BAT. It is also worth noting that humans have two different types of fats, brown adipose tissue and white adipose tissue. The main difference between the two is that the brown adipose tissue is used for production of heat, while white adipose tissue is used for energy storage. Heat production occurs in brown adipose tissue of humans and hibernating animals, but the amount of brown fat in humans drops with age, since it is mainly needed for newborns to reduce the risk of hypothermia right after birth. Uncoupling protein, UCP, is expressed in BAT. UCP is a protein channel in the inner membrane that uncouples oxidative phosphorylation and electron transport by allowing proteins to enter the matrix without ATP being synthesized. Instead, the transfer of protons into the mitochondrial matrix is affiliated with heat production. Uncoupling leads to elimination of the feedback inhibition and resulting in the continued synthesis of reducing equivalents through the oxidation of fuels such as carbohydrates and fats. Based on this mechanism, researchers are looking into upregulating BAT activity as potential weight loss therapy. Increased use of UCP can result in increased levels of oxidation of fatty acids and lead to weight loss. This leads us to a research question. Can inducing BAT activity via UCP1 provide a potential weight loss treatment? BAT is induced by cold temperatures and diet. For example, exposure to temperatures lower than 4 degrees Celsius has shown to increase metabolic rate, while starvation has shown to decrease metabolic rate. The sympathetic nervous system is the main effector component of this response to the brain. When the sympathetic nerves are activated via cold induction, a neurotransmitter, norepinephrine, is released. Norepinephrine interacts with the beta-3 adrenergic receptor, beta-3A receptor, on brown adipose tissue and activates adenylose cyclase. Adenylose cyclase will then convert ATP to cyclic AMP, which is CAMP, which activates protein kinase A, which is PKA. PKA will phosphorylate triacylglycerol-lipase to activate it, and lipase will break down triglycerides to free fatty acids, thus activating UCP1. Based on this mechanism, researchers are exploring the use of sympathomimetic agents, such as beta-A receptor agonists, to increase energy expenditure. This diagram is based on reference 1.
The following study from Enerbäck et al. Examined the role UCP has in the regulation of body heat by targeted inactivation of the gene encoding it. The study concluded that gene knockout mice lacking UCP1 demonstrate cold sensitivity that is usually not seen in wild-type mice containing the UCP gene. In this section, we will refer to wild-type mice as mice having 2 intact copies of the UCP gene, denoted by ++. Plus-minus refers to mice with 1 intact copy of UCP, and negative refers to UCP deficient or knockout mice. I'll provide a description for how the knockout was created in this experiment. The UCP gene was inactivated by homologous recombination in embryonic stem cells using a targeting vector. The BAMH1 and BG3 fragment was replaced with NEO-R gene, which completely removed exon 2 and parts of exon 3 in the UCP gene. This process deleted an essential membrane-spanning domain, effectively disrupting the UCP gene. The figure shows the restriction enzyme sites and the exons on the UCP gene before recombination, seen above, and after recombination, seen below. Next, a northern blot analysis and immunoblot of UCP were performed, which discovered the absence of UCP mRNA and immunoreactive UCP protein in knockout mice. Mice with 1 intact copy of the gene produced UCP mRNA at levels comparable to wild-type mice, as expected. No differences were seen in the levels of transcripts from mitochondrial localized cytochrome oxidase or the adipocyte-specific AP2 protein. In addition, there is a complete absence of UCP protein in UCP-deficient mice, as seen in the immunoblot. These 2 experiments confirm the loss of function of the UCP gene as a result of recombination. In the next experiment, they exposed 41 UCP-deficient mice to cold temperatures at 5 degrees Celsius to investigate the effects of UCP deficiency on thermal regulation. The mice sample consists of both males and females, who differed in age from 27 to 85 days old. The unfilled circles represent the males, the filled circles represent the females. If the mice lost 10 degrees of body heat, they were classified as sensitive to cold. If less than 10 degrees of body heat were lost after 10 hours in the cold, the mice were classified as resistant to cold. 35 out of 41 UCP deficient mice were sensitive to cold, compared to 6 who were resistant. Within the 35 cold sensitive mice, the onset of temperature loss varied from 1.5 to 9.5 hours. Sex and age was not a factor in cold sensitivity or resistance. For comparison, all 10 heterozygotic mice were resistant to cold.
Our reference 3 paper provided significant evidence on how increasing BAT through cold exposure controls triglyceride metabolism. They specifically looked at how triglyceride-rich lipoproteins transport in the bloodstream and how BAT could increase the liberation of fatty acids by increasing the action of lipoprotein lipase. The study specifically wanted to investigate the role of lipoproteins in delivering energy for BAT. Furthermore, they wanted to see if increasing triglyceride clearance through cold exposure would also lead to weight loss in both lean and obese mice. Before looking at the results, we need to understand how they measured blood plasma concentrations of triglycerides and fatty acids. So here we have our triglyceride lipoprotein complex. This is our phospholipid bilayer with cholesterol embedded in between. Then we have our fatty acids and triglycerides. They were able to follow lipoprotein and fatty acid uptake together by embedding both H-triolein and hydrophobic superpragmatic iron oxide, aka SBIO, nanocrystals into the core of triglyceride particles. Upon lipoprotein lipase breakdown, H-trioleate was released while FPSIO nanocrystals remain within the triglyceride core. These figures showed that cold mice had significantly faster H-triolein and Fe-SBIO clearance. This indicated that colder mice had lower concentrations of triglyceride flowing in the bloodstream, and that the fatty acids were being broken down through lipolysis and being absorbed. They're absorbed by peripheral organs such as the muscle and adipose tissue. As a side note, any cholesterol release would be absorbed by the liver, so the breakdown would not lead to high cholesterol levels. To link this triglyceride activation to BAT, they observed the organ distribution of H-triolein and Fe-SBIO by removing the rat's organ and measuring counts per minute of radioactivity. These results showed that accelerated clearance was mediated by BAT in cold mice versus control. They showed that cold exposure substantially increased lipid uptake through BAT, but seemed to reduce liver triglyceride uptake. This indicates that cold exposure shifted the clearance of lipoproteins from liver to BAT, demonstrating that BAT accelerates triglyceride clearance and is a major target for triglyceride uptake. Now they wanted to investigate whether increasing triglyceride clearance in the blood would decrease weight loss in obese mice. They had 4 types of mice to investigate: lean control and lean cold, which were both fed normal chow diets, while the obese control and obese cold had a high-fat diet to induce obesity. They had 10 mice per group. Observing the weight, they noticed that obese mice lost significantly more weight than control. However, both groups lost weight nonetheless. However, to test the significance, they looked at the percent weight loss, and here in actuality, the reduction in weight is similar between both groups. Using the same method as before, they measured lipid uptake in different rat groups. These results indicated that under cold exposure, BAT activity was the same between the obese and the lean group. However, in the control obese group, there was a higher triglyceride clearance compared to the control lean group. This is likely due to the fact that the obese mice were on a high-fat diet, so they utilized triglycerides more often for energy use. Using the same method as before, they wanted to observe organ distribution of H-triolein and Fe-SBIO in the 4 different rat groups. So under cold exposure, lean and obese mice had increased lipid uptake, which was mediated by BAT. They were also less reliant on the liver for lipid uptake compared to the control group. Together, all these results indicate that increasing BAT activity through cold exposure led to more triglyceride clearance. By increasing this clearance, we can induce weight loss, which would be a beneficial effect for an obese human patient. That is, if we can find a way to induce BAT without freezing them to death. Go.
A study conducted by Cypess et al. Aimed to examine if mirabegron, a beta-3 adrenergic receptor agonist, could activate human BAT, thereby increasing energy expenditure as a potential treatment for obesity. Beta-3 adrenergic receptor agonists have been seen to stimulate rodent bat, but this activity has previously never been demonstrated in humans before. Other beta-3 adrenergic receptor agonists have shown promise in their ability to increase glucose disposal, decrease fats in the blood, and increase resting metabolic rate, but none of these drugs have been approved for the treatment of obesity because of their poor oral bioavailability and substantial cross-reactivity with the beta-1 adrenergic receptor, which is primarily expressed in cardiovascular tissue, causing undesirable cardio effects. Mirabegron, however, has been approved for the treatment of overactive bladder at a dosage of 50 milligrams. It has satisfactory bioavailability as well as a proven higher binding affinity for the human B3AR receptor, meaning less off-target binding to the beta-1 adrenergic receptor and therefore fewer cardiovascular side effects compared with other members of its class. This study screened 15 participants for BAT activity through cold exposure, but only 12 had detectable BAT activity and could continue. From the cold exposure, the 12 subjects showed increases in resting metabolic rate, systolic and diastolic blood pressure, and a decrease in heart rate, consistent to what has been reported in the literature before. BAT metabolic activity was measured using prospective 18F fluorodeoxyglucose PET-CT imaging. A dosage of 200 milligrams of mirabegron was used because it has a higher efficacy for reducing the symptoms of overactive bladder than the standard dose, and this was because it was due to the limitation of only being able to do 3 such scans on each subject safely. As you can see in this figure, the principal sites of detectable BAT were the cervical, supraclavicular, axillary, adipose tissue depots, but glucose uptake was also even seen in the paraspinal and periphery regions. BAT activity significantly increased compared to the placebo overall, as you can see in this figure, and all 12 subjects' BAT glucose uptake was significantly higher after treatment with mirabegron. These figures show that compared to placebo, mirabegron also significantly increased RMR, heart rate, and systolic blood pressure, but not diastolic blood pressure. This drug-induced stimulation of the cardiovascular system was considerably lower than what has been reported for other broadly acting members of the B3AR drug class, which is promising in itself. There was also a positive relationship between changes in RMR associated with BAT activity in response to mirabegron, and not the placebo. The higher a subject's body fat, the greater the change in RMR, and therefore more BAT activity. There were also no associations between changes in heart rate or blood pressure. This again shows the promise that mirabegron could have for combating obesity. At the 200 milligram dose used in the study, the maximal energy expenditure was 200 kilocalories a day, and this leads to a predicted weight loss of 5 kilograms in the first year and 10 kilograms by the end of 3 years. However, the effects of mirabegron dosage was only examined for 1 day, and 200 kilocalories a day was the peak and not sustained throughout the day. All subjects in the study were young, lean, and healthy males, and as such, the efficacy of mirabegron needs to be evaluated in women and other demographics. Finally, a dosage of 200 milligrams is much higher than necessary to selectively activate only the beta-3 adrenergic receptor, and so there was some binding to the beta-1 adrenergic receptor with resulting tachycardia and cardiovascular effects. Therefore, an appropriate dosage size as well as long-term studies must be conducted to establish long-term safety. In summary, this study shows that the beta-3 receptor agonist mirabegron accurately stimulates BAT thermogenesis and increases resting metabolic rate, and since mirabegron is on the market, it could very likely serve as a potential treatment for obesity.
Capsaicinoids are small molecules derived from chili peppers. They are known to have a wide range of physiological effects, including the increase of metabolic rate. Although their effects are variable, several companies already offer capsaicinoid-based weight loss supplements. Recent research has revealed a possible mechanism through which capsaicin alters metabolic rate, as well as reasons why the effects of capsaicinoids are not always consistent between individuals. Transient receptor potential vanilloid type 1 channels, TRPV1 channels for short, are protein channels expressed throughout the sensory nervous system, including the gastrointestinal tract. Capsaicinoids are a known agonist of TRPV1 channels, and this raises the question of whether TRPV1 channels can be targeted to activate BAT. A recent study on rats yields evidence supporting this idea. This figure shows, from reference 5, sympathetic nerve activity at 4 different time points after a weight-dependent intragastric dose of the capsaicinoid capsiate. After releasing capsiate directly into the forestomach, researchers measured the activity of the sympathetic nerve innervating BAT. A large spike in activity was observed 3 hours after administration, and this effect was seen in all 6 rats tested. It is also important to note that blood pressure and heart rate did not change after capsiate administration. This indicates that capsaicin likely did not have any other significant effects on the sympathetic nervous system. To confirm that the increase in BAT sympathetic activity corresponds with increased thermogenesis, BAT temperature was monitored after capsaicin administration. This figure from reference 5 shows that capsaicin caused a significant increase in BAT temperature. Treatment with vehicle, or capsaicin and ruthenium red, a non-competitive inhibitor of TRPV1, did not yield the same temperature increase. Taken together, these results indicate that capsaicin in the GI tract can significantly increase BAT temperature. To further elucidate the pathway connecting TRPV1 with BAT, BAT sympathetic nerve activity was measured after capsaicin administration in 2 groups of rats. One group had the GI branch of their vagus nerve severed, while the other group was unchanged. The GI branch of the vagus nerve is responsible for carrying nervous signals between the GI tract and the central nervous system. The purpose of the vagotomy was to test whether the vagus nerve is involved in the capsinoid BAT activation pathway. This figure, also from reference 5, shows that capsinoids didn't change BAT sympathetic nerve activity in the vagotomy group, indicating that the gastric branch of the vagus nerve is likely required for capsaicin to mediate its effect. Overall, the evidence from this study demonstrates that capsaicin binding TRPV1 in afferent nerves of the GI tract triggers a nervous response that results in increased BAT thermogenesis. Although the previous study presents a viable mechanism for the activation of BAT by capsinoids, it does not explain why responses to capsinoids vary so much between subjects. This problem was addressed by a different study investigating the impact of capsinoids on healthy adult males. The group was further divided into those with active BAT tissue and those without. To determine which subjects had active BAT, the subjects fasted for 6 to 12 hours, entered a cold room for 1 hour, then were given an IV infusion of 18-fluorodeoxyglucose. The uptake of 18-fluorodeoxyglucose in BAT regions was monitored by PET-CT scan, and the amount of radioactivity was used to define a threshold for active BAT. After determining which subjects had active BAT, the resting energy expenditure of each subject was estimated with a respiratory gas analyzer. The same measurement was then carried out after the administration of a capsule containing 1.5 milligrams of capsinoids. This figure from reference 6 shows the difference in measurements for each group, as well as the combined measurements. It is evident that the capsinoids had a much greater effect on the BAT-positive subjects. The underlying variation in BAT activity between subjects helps explain why capsinoids do not always increase metabolic rate significantly. In order for capsinoids to increase metabolic rate, at least in the short term, the subject must already have a significant amount of BAT. Although this may seem like a limitation, studies on mice have shown that long-term capsaicin administration can actually upregulate UCP1 levels in BAT. In this way, capsaicin may also be able to activate previously inactive BAT. To sum up, capsaicin's ability to activate BAT through the GI tract with minimal side effects on the rest of the sympathetic nervous system makes it a possible candidate for new weight loss treatments.
To summarize, UCP1 is responsible for uncoupling of oxidative phosphorylation in BAT, thereby producing heat and increasing metabolic rate. BAT can be activated by cold exposure as well as small molecules. Thorough research in rat models has shown that increasing BAT activity leads to weight loss, but this effect may be difficult to translate to human patients. This is because rats have higher levels of BAT when compared to humans. As a result, future weight loss treatments may be a challenge for all human patients as the amount of BAT varies between individuals.